DNA forms a double helix, kind of like a twisted ladder. This is the backbone that consists of deoxyribose phosphate. Two strands of nucleotides hold it together, A to T and G to C. Now, these two nucleotides are not created equal. For example, the G to C has 3 hydrogen bonds, which we're going to talk about in a minute, and the A to T only has 2. Well, if you've done basic elementary math, you know 3 is more than 2. And what that means is these bonds are harder to break, whereas the A to T are significantly easier. And that leads to something that we call TATA boxes, which are areas of TATA. These are regions in which DNA replication enzymes or other enzymes can come through and break more easily because there is a lower concentration of G and C. Also, G and C is one of the characteristics, or the concentration or ratio of G to C are some of the characteristics that we use to define different organisms. So they're usually the higher the concentration of GC, the more extreme environments they can reside in, because again, this is going to hold your DNA better together than the A to T. The strands are anti-parallel, which means you have one side going from 5' to 3' and one side going from 3' to 5'. And that prime I'm going to show you are the sugars. Well, it's the carbons number that the replication enzymes recognize. So the order of nitrogen-containing bases forms the genetic instructions of the organism. So here we have our 5' end, okay? So that's this carbon right here. And here we have our 3' end, and this is this carbon right here. Now, replication always occurs from 5' to 3'. We're going to talk a little bit about that and what that actually means in just a second. So here we have our 2 hydrogen bonds between our T and our A, and our 3 hydrogen bonds between our G and our C. One strand serves as the template, and what that means is it's called semi-conservative replication, which means we don't start from scratch and rewrite everything. We have a template that we start on. So we know that because there is an A on one side, that's the old half of the DNA, there's going to be a T on the other. We know because there's a G on one side, there's going to be a C on the other. And so if there's ever a nucleotide that's brought in incorrectly, we can look at that parent strand and say, oh, we made a mistake there. Let's go back. So the topoisomerase and gyrase relax the strands. So if you've ever tried to pull 2 strings apart, you probably noticed they started getting kinked and there were a lot of tensions. The topoisomerase and the gyrase cut the DNA to allow it to unwind a little bit and then reseal it. The helicase itself separates the strands, and that's where the replication fork begins. So here is our double helix, and right here would be where the topoisomerase and the gyrase cut the DNA to relax some of the tension that's being put on the helix by it separating. This is our replication fork, and you can see our nucleotides appropriately matching with their complementary hydrogen bonds. So this is the 5' daughter end. This is the 5' parental end. So what that means is you have replication occurring here from 3 to 5, from 5 to 3. We're going to talk about how that occurs in just a minute. DNA polymerase adds nucleotides to the growing DNA strand in a 5' to 3' direction. Now, we use an RNA primer to start synthesis. The RNA primer itself can only recognize the 5' end, which was shown to you in a previous slide. The leading strand, which is the one that is going in the correct direction from 5' to 3', is synthesized continuously because, hey, it's doing it correctly. The lagging strand is doing it wrong. Remember, each of these strands is anti-parallel, which means when you have a 5' to 3', you're going to have a 3' to 5', which means that side cannot be replicated in the correct fashion. So what happens is you do these short replication patterns and then sew them up together at the end. Let's show you a picture. So here you have the 5' carbon going to the 3', and replication here is going from 5' to 3'. So this is being replicated continuously with the DNA polymerase. Remember, an RNA primer sat down, started replicating, and then you have other proofreading proteins that are going there. These are the gyrase and the topoisomerases that are unwinding and relieving pressure from the helix. These are proteins designed to stabilize the two pieces of DNA, because if you've ever worked with double-sided tape, you know that things can get messy, so these are just sitting there going, all right. Now, this is where it gets tricky. Here you have a 5' to 3', remember, because this is the opposite of this top replication fork. But it still needs to go 5' to 3', but it's going in the opposite direction. So what we have is we have this little RNA primer, and then the DNA polymerase does these short replication patterns. And then after enough of the DNA is made, you get this DNA ligase that goes through and joins these strands together, or does a lot of kind of patchwork, if you will. So DNA replication. Energy for replication is supplied by the nucleotides. Hydrolysis of the two phosphate groups on the ATV is what provides the energy. So here we have our new strand and our template strand. Okay. And so as you have ATP right here, this allows for the sealing and use of the phosphate to the adenosine. Most bacterial replication is bidirectional, which means it's occurring simultaneously, going in opposite directions of the circle, because again, bacterial genomes are circular. Each offspring receives one copy of the DNA, and the replication is highly accurate due to the proofreading capability of DNA polymerase, which I mentioned previously. That proofreading function is of the utmost importance, because if proofreading is poor, there's going to be even a higher rate of mutation. So this is that bidirectional fashion. So you have a replication fork here and here, and you can see then they just kind of separate like two paired rings. Ribonucleic acid, again, single-stranded nucleotide, 5-carbon ribose sugar. In RNA contains a uracil instead of thymine. The reason for this is uracil has one less methyl group, so it's much cheaper to produce than your thiamine. The problem is it's relatively unstable. Well, we don't care really if RNA is unstable, because we can just make more, and we make a lot more RNA than we do DNA. So because of that, we need that energy cost savings. So you have several types of RNA. You have your ribosomal RNA, which makes up your ribosomes, your transfer RNA, which escorts amino acids to the ribosome to be included on the protein strand that you're working with. Then you have your messenger RNA, which carries the coded information from the DNA to the ribosomes. Transcription in prokaryotes, synthesis of complementary mRNA strands from a DNA template. Transcription begins when RNA polymerase binds to the promoter sequence of the DNA. Transcription again proceeds from 5' to 3'. only one of the two DNA strands is transcribed. Transcription stops when it reaches a terminator sequence. And so you have usually a stop codon or a terminator that lets you say, hey, the gene ends here. But when sometimes mutants occur, which may change the stop sequence, which means that you get an incomplete protein. Now, in humans, this creation of message or mRNA is done in the nucleus. And then that RNA, mRNA, is escorted out of the nucleus to the adjacent ribosomes in the endoplasmic reticulum, the rough endoplasmic reticulum. There are also ribosomes in your cytosol, but we'll talk about those more when we get to the viruses. Translation: mRNA is translated into a language of proteins. Codons require 3 nucleotides, okay, and they code for a particular amino acid. There are 61 sense codons that encode for about 20 amino acids. The genetic code involves degeneracy, meaning an amino acid is coded by several codons, which works in our favor. So, for example, if you had mutations in your UC, right? If it was UCU, UCC, UCA, and UCG, you would still have serine be produced, which is great, okay? Now, the problem here is if you're making tyrosine, right, you have UAC and UAU. That makes tyrosine. But if the mutation occurs and it is a UAA, it stops immediately, which means you'll have an incomplete protein, which could reduce functionality or make it non-functional altogether. Now, you only have one start codon, AUG. Now, OG, the way I remember this is inauguration. It's when you have a new president come in, right? It's their inauguration. So AUG is OG. Translation of mRNA begins at the start codon, OG, and translation ends at the stop codons, of which there are 3. I expect you to know both the start and the stop codon. Codons are read sequentially, which means in order. tRNA molecules transport the required amino acid to the gene. Now, if you do not have the required amino acid, that protein does not get made. And this is where a concern in vegan diets comes in. If you do not have appropriate amino acids in your vegan diet, some of your genes will not get expressed. When some of those genes are not expressed, that can lead to a deficiency, which may lead to cancer. It's very easy to get a balanced diet of amino acids as a vegetarian that still eats animal byproducts such as milk or eggs, because those have all the essential amino acids for life. So this is just a diagram of the tRNA docking here at the bind site. The complementary nucleotides are here, which then drops off the amino acid to be linked to the longer strain. Again, you have the peptide bond forming, and you have new amino acids constantly joining your new long protein. In bacteria, translation can begin before transcription is complete, because remember, bacteria are a big ball of ribosomes. So you can have it coming right off the nucleotide, and the ribosome is just right there. So it allows for a very efficient process. Now, the transcription in eukaryotes is a little bit different. We're going to talk about that in our next video.